Good morning, everybody. In the previous sections, we have discussed the role of elements and the growth factor in microbial growth. And next, we have also discussed the classification of media based on consistency and based on ingredients. Now, we are going to discuss the classification of media on oxygen requirement. Microorganisms have requirement for growth depending on oxygen requirement. As per example, aerobic media, as per example, anaerobic media. So in aerobic media, it is the media, it is easy to cultivate microbes on solid media. And the growth occurs by keeping the culture in incubator. And it shows the growth of non-fastidious microorganism. Not the fastidious, it is non-fastidious. So aerobic media, in this media, it is easy to cultivate microbes on solid media. The growth occurs by keeping the culture in indicator, and it shows the growth of non-fastidious microorganism. As per example of aerobic media are liquid media and solid media, like peptone water, 1% peptone and 0.5% NaCl and 100 ml water. Peptone water is a media where you can use for growth of aerobic microorganisms. And what is the composition of peptone water? Is 1% peptone, 0.5% NaCl and 100 ml water. Nutrient agar and nutrient broth plus 2% agar. These are the examples of aerobic media. But what is aerobic media? It is easy to cultivate microbes on solid media. The growth occurs by keeping the culture in incubator. Now, what is anaerobic media? The anaerobic media cultivates anaerobic bacteria at low oxygens. Here like the difference from aerobic media and anaerobic media. Aerobic media or aerobic organism requires oxygens, a large amount of oxygens. And anaerobic media, the media cultivates anaerobic bacteria at low oxygen or no oxygens. So the media cultivates anaerobic bacteria at low oxygens. So reducing oxygen reduction potentials. Anaerobic media contain extra nutrients like vitamin K, hemine, and oxygen, and that get reduced by a physical or chemical processes. The addition of glucose, thioglycolate, ascorbic acid, cysteine, or iron findings added to cause the media to reduce. And the medium is boiled in water bath to force out dissolved oxygen and packed with sterile paraffin. So here like the difference between aerobic media and anaerobic media. As per example of anaerobic media is RCM, Robertson cooked media meat, isolation for Clostridium species, because Clostridium is a anaerobic bacteria. So for isolations for culturing of the Clostridium, you need to use RCM. And what is RCM? Is a Robertson cooked meat media. Now, another example is thioglycolate broth. It has sodium glycolate and that maintain low oxygen. Low oxygen. So, is it clear that what is aerobic media and what is anaerobic media? What are the purpose of use of aerobic media and what are the examples of aerobic media and anaerobic media? Now, we're going to discuss types of special purpose media. These are not in the classification, but we need to understand the special purpose for media. So the name justified the significance, the special purpose. Like assay media, the media assay vitamins, the media assay for amino acid, the media assay for antibiotics. As we all know the importance of vitamins, as we all know the importance of amino acid, as we all know importance of antibiotic. For example, antibiotic sensitivity test, the media used here, Muller's Hinton's agar has 1.7% agar for better diffusions of antibiotics, and it also contains starch with absorbed toxins released by bacteria. And in this media plate, zone of inhibition is seen around the antibiotics. So that means whenever you are try to assay the antibiotic, so you need to use this assay media. Now next one, minimal media. What is the principle of minimal media? The minimal media is a defined medium with different compositions depending on microorganism culture, and it contains a carbon source like sugar or succinate and inorganic salts like magnesium, nitrogen, sulfur, phosphorus. Carbon is the source of energy. Magnesium and ammonium salt are the source of iron for metabolism stimulations, and phosphate is a buffering agent. And the growth comparison of microbe culture and mutant forms, minimal media and supplementary minimal media allow the differentiations of wild type and mutant cells. So this is the use of minimal media. And what is the as per example of use of minimal media? The selection of recombinants for growth of wild type microorganisms. So we have already learned assay media and minimal media. Now, what is the fermentation media? This is also special media and it will use for special purpose. The media is for optimum growth for microorganisms. Fermentation media produce high yield of the product. Media provide energy and nutrient for growth, and medium gives the substrate for the synthesis of the product in these fermentations. Fermentation media contain major and minor component, like major element, like minor element. So as per here is major components and minor components. In major components, carbon and nitrogen used here for the energy, and for minor component, this contain inorganic salt, this contain vitamin, this contain inorganic salt, this contain vitamins, buffer, anti-foaming agent, dissolve oxygen, gases, growth inhibitors, enzymes. These are all used as minor element components. The nutrient in the fermentation media depend on organisms and type of fermentation process. The nutrient in fermentation media depends on organisms and types of fermentation process. For two use, as for example, growth media, it has low nutrient and creates raw material for further fermentations. And fermentation media, it has high nutrient and creates end product. As for examples, the yeast requires 1% carbon, but fermentation of alcohol depend 12 to 13% of carbon in the medium. So here like the difference. Whenever yeast grow in the medium, not for fermentation purpose, it requires only 1% carbon. But whenever you are using yeast for fermentation process, fermentation purpose, there need demand of 12 to 13% carbon in the medium. So here like the significance. So what is the role of fermentation media? The media has a high level of nutrients, microorganism, and optimum conditions. During the incubation period, under optimum condition, microorganisms undergo metabolisms, and fermentations organisms become hyperactive due to nutrients being in high quantities, and the result is nutrient getting consumed and media partially degraded. And the waste effluent is the output product. And the death of the cells occurs if the substrate level reaches the inhibitory concentrations, and excess substrate cause them to inhibit vital enzymes, and excess substrate increase osmotic pressure and disturb enzymatic activity in cells. Microbes release excess substrate as partially digest fermentation media and convert it into insoluble inert compound as reserve food, and which is harmless to cells. As for example, yeast extract, as for example, beef extract, as for example, YPD, as for example, BMGY. This is the role of fermentation media. So now next we will discuss the application of culture media and limitation of culture media. So we have discussed different types of media previously, different types of culture media and different types of special media too. But what are the application of culture media? We need to know what is the application of culture media. The culture media, the culture can be used for the media can be used for culture microbes. Number one. Number two, identify the cause of infections. Number three, identify and characterize the microorganisms. Number four, to isolate pure culture. Number 5, to store the culture stock. Number 6, to observe the biochemical reactions. Number 7, to test microbial contamination in any sample. Number 8, to check antimicrobial agent and preservative effect. Number 9, to observe microbe colony type, its color, shape, and cause. Number 10 is to differentiate between different colonies. Number 11, to create antigens for laboratory use. Number 12, the estimate viable count. Number 13, to test antibiotic sensitivity. There is a lot of applications of the culture media. So this is the importance of culture media. But culture media, as beside its application and beside its importance, it has some limitations. It has some limitations. We need to know the limitation of culture media. Number one, the most important limitation is risk of cross contaminations. Number two, high skill required for optimal result. And number three, increased drying out of the media can occur. These are the limitation of culture media. This is very important to know whether you are using culture media for culturing the microbes. You need to know the applications as well as limitations. So here is a table, is a table. The table is examples of culture media for microorganism with simple and demanding nutritional requirement. As we will discuss previously, the different culture media compositions and its applications. Here also a table here also represented in a table. Like in the left side, it is define the culture media for Escherichia coli. Escherichia coli or E. coli is a very popular microorganisms for its wide applications for research purpose, for application purpose. So here the different compositions like K2HPO4 in need 7 gram, KH2PO4 2 gram, NH4 2SO4 1 gram, MgSO4 0.1 gram, calcium chloride 0.02 gram, glucose 4 to 10 gram, and it needs trace element like iron, cobalt, manganese, nickel, etc. And it needs dissolved water, and pH will be the 7. This is the culture medium for Escherichia coli, and it is also called defined medium because all the ingredients here actually known. Next one, the defined culture media for Leuconostoc mesenteries, the different organisms, and you can compare the here the culture media of Escherichia coli and culture media on for Leuconostoc. You can see here KH2PO4 is 0.6 gram, KH2PO4 is 0.6 gram, and K2HPO4 also 0.6 gram, and NH4Cl3 is NH4Cl is 3 gram, MgSO4 0.1 gram, and glucose is 25 gram, and it requires sodium acetate 25 gram, and it's required amino acid, purine, vitamins, and trace element, and also pH 7. But here you can find some similarities, like pH is same, but the ingredients is different because this ingredient is very specific for leuconostoc, and as you compare to Escherichia coli. And the next one, as you compare complex culture media for other complex culture media for either E. coli or mesenteries, leuconostoc mesenteries, you can use glucose 15 gram, you can use yeast extract 5 gram, you can use peptone 5 gram, KH2PO4 2 gram, distilled water 1000 ml, and pH will be 7, and the media color will be like yellowish. And another examples is defined culture media for Thiobacillus thioperas is here. KH2PO4 will be 0.5 gram, and NH4Cl will be 0.5 gram, MgSO4 0.1 gram, calcium chloride 0.05 gram, KCl 0.5 gram, and is requires trace element and also carbon CO2 from air. So we can understand here that for here it is defined by all are defined media, but the composition is different because the different microorganism has been used. They have different requirement for their growth. So we have discussed previously that the elements, importance of elements and their growth and their different media. and their compositions, their applications, their limitations of culture media. But now we will learn technically how can you transfer a liquid cultures in the laboratory. First, here the slide, there are two pictures. The upper part is express transferring a culture media, and lower part it discuss about streaking up petri plate. This is a two process, the laboratory process by which you can culture a liquid culture, transferring a liquid culture, and as well as you can culture a microorganism by streaking method. This is the tool actually. So we will discuss the first pictures, the first upper panel of the pictures. Yeah, you can see here is 1, 2, 3, 4, 5, 6, 6 steps is mentioned. And step 1 is you need to flaming the loop for sterilize it. Next one, you need to tube cap need to be removed. Next, flaming the tube tip sterilize the surface. Number 4, only sterilized portion of the loop enters the tube. Number 5, the tube is again reflamed, and then tube is recapped, and then steps 2 to 4 repeat with the tube of fresh medium, and the loop is then restricted in the flame. And this is the procedures. This is the 6-step procedures. You can transfer a culture medium from one tube to another tube or one media to another media by this process. And the lower panel is streaking a petri plate. Here also the 3 steps defined. As previously we already learned in upper panel that how can you transfer a liquid culture, right? So the first step here is loop is sterilized and loop full of inoculum is removed from the tube. Then you have a culture media, solid media, you have a plate, right? And you need to subsequent streaks are at angles the first streaks, and this is the streaking method. And initial streak is worked is well and in one corner of the plate, and if you incubate this plate for a certain time like 12 to 20 hours or overnight, you will see the colonies on the plate as per pictures in the left side in the lower panels. And this is the isolated colonies, the creamy white appearance of the colonies is the isolated colonies at the end of the streak. And this and we can see also the confluency of the growth. There's a lot of growth, and the growth look like lawn. So here you can find single colony as well as lawn, because where the microorganism population is very high, here you can find confluent growth, and where microorganisms is single, because this colony is actually coming from a single microorganism. The microorganism grow gradually, and whenever a single population, single bacterium gradually grow, it will at the end it will form a colony. Colony, it's a isolated single colony. So this is the appearance of oil stick plate after incubations. Show colonies on the bacterium, and this is the example of Micrococcus luteins on a blood agar plate. So we are already, so this slide is expressing how can you transfer a liquid culture and sticking a plate. Next, so we will learn how can microorganism grow like media, or how can transfer a culture medium, how can obtain a colony. Okay, or we already discussed previously that microorganism can be grow in liquid culture too. But now we going to discuss how can you count the microorganism? How can you count the microorganisms? Here is the example of direct microscopic counting procedure using the Petrov-Hauser counting chamber. So in the picture, in the slide pictures, you are seeing the left side is like a there is a stage, and where look at that, these use cover slips, and below materials is ridges that support the cover slip actually. And here is actually the chamber, and that chamber, if you add some liquid grown cultures, you need to add sample. So care must be taken not to allow the overflow, because space between the cover slip and the slide is 0.02 mm. So you can add the culture in between cover slip and slide. And after taking the cultures to the slide, you need to cover with the cover slips. And there is also a space between the cover slip and slide is 0.02 millimeter. Okay. And then whole grid has is a grid like appearance, and whole grid has 25 large squares. A total area is 1 millimeter square, and the total volume of 0.02 millimeter cube. Then if you see under the microscope, whenever it is like uh, microscope, you will see the microorganisms. And here the second pictures is the microscopic observations. All the cells are counted in large squares, 16 small squares actually, and 12 cells. 60. If you see, the last square is containing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 cells. So in practice, several large square are counted and number will be average. And how can you calculate from this? How can you calculate the total number, the microorganism present in this liquid media? So to calculate the number per milliliter of the samples, we can use this mathematical form equation like there is 12 cells and 25 large square. So you need to multiply 12 cells into 25 large square into 50 into 10 to the power 3. Okay, so this is the mathematical formula by which you can count the number of bacteria, and you can count average number of bacteria also per ml. So this is the and this is the phase contrast microscope, and you need to use a phase contrast microscope. And this my phase contrast microscope is typically used to count the cells to avoid necessary for staining. Okay, so this is the process of direct microscopic counting by Petrov Hauser counter. Now we will learn previously that you can count the microorganism. Okay, but how can you count the viable cells on culture media? How can you count the viable cells on culture media? Here we will going to discuss the methods for the viable count. The upper panel is the spread plate technique and lower panel is a pour plate technique. There are two technique has been used for viable count. Number one is spread plate technique. Number two is pour plate technique. In the spread plate technique, you need to use samples is pipette onto the surface of agar plate, like as for example 0.1 ml or less, like 30 microliter or as per example like 50 microliter, 60 microliter, you can use as per requirement. And after you pipetting the samples onto the agar plate, the sample is sprayed over the agar surface using sterile glass spreader, and you need to incubate the plate to observe or to get visible colonies. And typical and you can see the typical colonies like left side panel, the surface colonies. And whatever, this is the spread plate method by which you can obtain the colonies. And this is the methods for viable count. You can count the colony number from this plate. Next one, the pour plate method. The pour plate method is a very simple, and a sample is pipetting onto a sterile plate. Then sterile media is added and mixed well to the inoculum. You need to add sterile media added and mixed oil inoculum. Solid, you need to for solidifications and incubations, and you will get typical colonies. So only the surface colonies from the spread plate technique. By contrast, in the pour plate method, colonies form within the agar as well as on the surface. But on the far right, photos of the colonies, Escherichia coli form the cells plate by spread plate top or pour plate bottom. This is the overall procedure. This is a procedure by spread plate technique and pour plate technique to method for viable cells. And another technique we will learn, the procedure for viable cell count using serial dilutions of the sample and pour plate method. This having applications widely nowadays for in research as well as industry purpose, as well as many application uh, like to isolate organisms from soils, to isolate organisms from water source. This serial dilution is a technique that will be used for the isolation of colonies. So how can we isolate colonies by serial dilution? Look at that. The sample to be counted. How? So in the middle part, we are using some test tubes, and and in the upper panel we can see the culture conicals containing cultures. So if you take 1 ml from the cultures into 9 ml broth, the dilution will be 1 by 10th. So you can say its a dilution will be 10 to the power minus 1. And again, if I take 1 ml from this diluted cultures, that is from 10 to the power minus 1 to fresh one in 1 ml to in 9 ml, then it will be 10 to the power minus 2. Again, if you take 1 ml from 10 to the power minus 2 to fresh tube 9 ml, then it will be 10 to the power minus 3. So here, like, if you again take 1 ml from 10 to the minus 3 and into 9 ml broth, it will be 10 to the minus 4, and 10 to the minus 5 and 10 to the minus 6 can be made by this process. And this is actually called serial dilution, because a series is maintaining serially cultures is taking from the previous diluted cultures, and the dilution factor can increase from 10 to the minus 1 to 10 to the minus 2, 10 to the minus 2 to 10 to the minus 3, 10 to the minus 3 to 4, 4 to 5, and 5 to 6. And then you need to take culture from each dilutions, and you need to plate it in the medium. So you will find too many colonies in the lower dilution plate, like in 10 to the minus 1 and 10 to the minus 2. But if you but too many colonies, and these colonies will be uncountable, because there are large number of colonies in the plate media, but where you can find the less number of colonies in 10 to the power 6 and 10 to the power 5, very less number of colonies, because they are highly diluted. But you can count comfortably from 10 to the power minus 3 and 10 to the minus 4 plate, right? And whenever you are counting the colonies, the colony numbers must be multiplied by the dilution factor to obtain the total cell number. As for example, from 10 to the power 3 dilutions, you are finding 159 colonies, and these 159 colonies, the plate count you getting 159 colonies, and the dilution factor is 10 to the power 3. So how many number of cells will be there in 159 into 10 to the power 3 is equal to 1.59 into 10 to the power 5. So this is the cells. It's called actually colony forming unit, CFU, per milliliter of the original samples. So this is the overall procedure for viable counting using serial dilution of the samples and the pour plate method. And the sterile liquid used here for making dilutions can simply be water, or it can simply be the media, as for example LB broth. But a solution of mineral salt or actual growth media may yield in a higher recovery, and the dilution factor is reciprocal of the dilutions. So this is the procedure of serial dilution method to obtain colonies and to count the colony number. And the next we'll discuss the now we'll already learn how to count the colony, how to observe the colony, but we need to count the bacterial cell growth, and that growth can be quantified from turbidimetric measurement, turbidity. And this turbidimetric measurement can be done by the instrument. Here you can see the pictures is spectrophotometer. Spectrophotometer based on Lambert's Beer's law, where light source has been used, and this light will be absorbed by the microorganism, and these observations can be recorded in spectrophotometer. And these observations will be expressed in terms of optical density. Here we can see as per examples here wavelength. If you are using the wavelength 540 nanometers for that bacterial growth, you are seeing the OD value is 0.32. OD is nothing but a optical density, is a measurement of turbidity. Turbidity. In the lower panel, you can see that at OD 540, the cell, there is like appearance like very faint of the bacteria, and upon time, look at that, the cell number increase and the turbidity become increase. And so if you grow the microorganisms in a time dependent manner, we can plot the growth as per optical density and time. In the graph actually shows here the two organism A and organism B. Here the and their growth and their optical density, it has been reflecting here on time period. Okay, so this is the procedure by you can measure the microbial cell number from turbidity. We will learn next how can you uh, count the colony forming unit or bacterial cell number, and at a particular time period with corresponding to optical density, and that will be reflect in curve, and that curve will be called growth curve and typical growth curve. So bacteria always maintain a typical growth curve. We will learn next the what is growth curve, and how what are the phases in the growth curve, and how its significance, and mm, how can we get a growth curve in any microorganism by culturing. Thank you. Thank you for the listening.